Black Death is still affecting the human immune system. The Flagg Bruel fit in East Smithfield, London, researchers analyzed the DNA from the remains of people extracted from the feet, which were used for massive burials in 1348 and 1349 amid a flag outbreak in the city. CNN, the Black Death, the world's most devastating flag outbreak, killed half of medieval Europe's population in the space of 7 years in the 14th century, shifting the course of human history. But what about the survivors of what remains the single greatest mortality event ever recorded? New research published Wednesday in the journal Nature suggests that it was the modern luck that determined who lived and who died. Analysis of centuries old DNA from both the victims and survivors of the Black Death has identified the key genetic differences that helped the people survive the plague. According to a study published in the journal Nature, these genetic differences continue to shape human immune systems today, with genes that once compelled protection against the plague now linked to the greater vulnerability to autoimmune diseases such as Crohn's and rheumatoid arthritis, the study said. We are the descendant of those that survived past the pandemics, and understanding the evolutionary mechanisms that contributed to our survivors is not only important from the scientific viewpoint, but can also inform on the mechanisms and genetic determinants of present day susceptibilities to disease, said study co-author Luis Barreiro, a professor of genetic medicine at University of Chicago, via email. Flagellate specimens. The 7-year study involved the extraction of DNA isolated from three different groups of skeletal remains unearthed in London and Denmark. Flagell victims, those who died before the Black Death and those who died between 10 and 100 years after the flag struck. More than 300 samples came from London, a city hit particularly hard by the flag, including from individuals buried in the East Smithfield flag pit used for massive burials at the height of the outbreak in 1348 to 1349. Another 198 samples were taken from human remains buried in 5 locations in Denmark. DNA was extracted from dentin in the root of individuals' teeth, and researchers were also able to check for the presence of Yersinia pestis, the bacterium that caused the plague. They that research they then searched for signs of genetic adoption to the disease. It's a long process, but in the end, we have the sequence of those genes from those people from before, during, and after the flag. And you can ask, do the genes one population carried look different than the ones another population carried? Said co-author Hendrik Poiner, the professor of anthropology at McMaster University in Hamilton, Ontario, in an email. The team pinpointed a variant of one particular gene known as ERAP2 that appears to have a strong association with the flag. But before the Black Death, the variant of the ERAP2 found to be protective to the flag was found in 40% of individuals included in the London study. After that Black Death, it was 50%. In Denmark, the percentile disparity was starker. It changed from about 45% of samples buried before the flag to 70% buried afterwards. The team don't yet know exactly why this variant compelled protection, but their lab experiment in cultured cells indicates that in people with the ERAP2 variant, an immune cell known as the macrophage provoked a very different response to Yersinia pestis. Barreiro explained. Macrophages from individuals with the variant are better able to kill the bacteria in lab experiment than from macrophages from individuals lacking it. We do not know if it still protects against the flagyr, given that the number of cases in present day populations is very low, but we speculated that it should, he said. It's also likely that the variant is beneficial against the other pathogens, although this wasn't part of the research. More than 300 samples came from the East Smithfield Flagyr Fit in London. Price of immunity. The downside to the variant is that it has been linked to the greater susceptibility to autoimmune disorders such as Crohn's disease, where the immune system becomes overactive. This suggests that populations that survived the Black Death pay the price, which is to have an immune system that increases our susceptibility to react against ourselves, Barreiro said. He said that it was unlikely that COVID-19 outbreak would shape our immune system in a similar way, largely because the disease predominantly kills people after their reproductive age, meaning it's unlikely genes that compel protection would be passed on to the next generation. This change in human genetic makeup occurring in the matter of these decades is also a rare example of rapid natural selection, said David Arnold, a professor in the University of Arizona's Department of Ecology and Evolutionary Biology, who was not involved in the research. The narrow time window from which samples was taken and the large number of samples analyzed are selling point of the study, he said in a commentary published alongside the study, allowing the authors to accurately date natural selection. Even though evolutionary biologists had previously wondered about the possibility of natural selection during the Black Death, proper investigation was not possible without the precise dating of many samples.